Welcome to the part 3 of the cell suspension culture. Cell suspension culture. See, the tissues and the cell culture they are cultured in an agitated fluid medium. So it will produce a suspension of a single cell and the cell clumps. There is a cell clumps of few to many cells. So this is called as a suspension culture. What is suspension culture? I'll just write over here. Tissues and the cells cultured in an agitated medium, liquid medium. That will produce a suspension of single cells and cells clump of few to many cells are called as suspension culture. So what is done over here is a culture can be inoculated in a liquid medium, and through continuous shaking, the cell suspension cultures are obtained. So we can say suspension culture grows much faster than the callus culture. And there are many different methods of suspension cultures which have been developed. They fall into two main types. The first is the batch culture, in which the cells are nurtured in a fixed volume. Fixed volume of medium until the growth eases. And second is the continuous culture, in which the cell growth is maintained by continuous replenishment of sterile nutrient media. So how it is done? We select a plant, organ or a tissue, then it is rinsed and sterilized with the, we will rinse and sterilize the explant, cut and place the explants on a solid medium for 2 to 6 weeks. The callus culture is formed. Now chop or cut this callus and put it in a liquid medium for 1 or 2 weeks. The cell suspension culture is obtained. This is how it is done. The next culture which we will be studying is the organ culture. See, it is a general term for those type of culture in which an organized form of growth can be continuously maintained. Like it will include the aseptic isolation of definite structures like leaf primordia, immature flowers, fruits, and their growth in vitro. Differentiated plant organs can usually be grown in a culture without the loss of the integrity. So there are two types which we will be discussing is the determinate organs which are destined to have a defined size and shape, for example, leaves, flowers, fruits. And second is the indeterminate organ, where the growth is potentially unlimited राइट जैसे आपका एपिकल मेरिस्टम ऑफ द रूट्स हो गया या नॉन फ्लावरिंग शूट्स हो गया ठीक है सो इन द ऑर्गन कल्चर फर्स्ट इज द मेरिस्टम कल्चर so what is the characteristic of a meristematic cells they retain the power of the division long after the embryogenesis is over okay based upon its position in the plants there are three types of meristematic cells which are present that is apical intercalary and the lateral meristem apical meristem is found at the tip of the root stem so I can write meristems ke andar there are three types it is apical it is intercalary and the last is the lateral. Apical, it is found at the tips of the root. Stem or branches. So major role इसका क्या होता है? Increase the length of the plant. Usually जो culture है ना, अगर आप apical meristem का करते हो, particularly of the shoot apical meristem, it is called as a meristem culture. ठीक है? Meristem culture particularly if the culture of shoot apical meristem. This is called as meristem culture. Now, what are the advantages? Why we are using the meristem culture? So, major advantage of using the meristem culture is that it provides I'm writing the advantages. It provides clonal propagation with maximum genetic stability. And second is the production of virus-free plants. See, the production of virus-free plants Because since the infected plant meristem, typically they're either near or they are either nearly or totally virus free. So the reason proposed for the escape of the meristem from the virus invasions are, why it is so, because the virus readily move in the plant body through the vascular system, which is absent in the meristem, right? Second is the high metabolic activity in the actively dividing meristem cells. And this does not allow the virus multiplication. Third is the virus inactivating system. In the plant body. If any has a higher activity in the meristem than in any other region, thus the meristem is protected from the infection. And the last is the reason why the meristem is not having a virus, because of the high endogenous oxygen levels in shoot apex that may inhibit the virus multiplication. Next culture which I'm doing is the embryo culture. So embryo culture involves the isolating an immature or a mature zygotic embryo aseptically and growing it on an aseptic nutrient medium with the goal of obtaining a viable plant. So there are two types of embryo culture. Immature embryo culture and another is the mature embryo culture. So Hanning was the first person in 1904 who obtained a viable plant from aseptically isolated mature embryo, which was grown on a mineral salt medium supplemented with sugar. Later, in 1924, Dietrich cultured mature and immature embryos of various plant species and reported that the mature embryos grew immediately. So, like, so this culture of embryo culture is widely used for growing plants in which the failure of endosperm to develop development they cause embryo abortions. ठीक है, वहां पे इसको use किया जाता है. तो embryo rescues this procedure. The artificial nutrient medium serve as a substitute for the endosperm and thereby allowing the embryo to continue to develop. So there is a technique called as the embryo rescue technique, which has been utilized to obtain a progeny from the intraspecific hybridization that do not normally produce a viable seed. So depending upon the organ culture, it can be a embryo rescue, may be referred to as an embryo, ovule, or an ovary culture. The most commonly used is the embryo rescue procedure, in which the embryo culture, in which the embryos are excised and they are placed directly onto a culture medium. So embryos are difficult to excise when they are very young or from the small seeded species. To prevent the damaging embryos during the excision process, they are sometimes cultured while still inside the ovule. So this technique is called as an ovule culture. In ovary or a pod culture, the entire ovary is placed into the culture. अब इसकी एप्लीकेशन कहाँ पे आती है? एप्लीकेशन number 1 is the overcoming the embryo abortions due to incompatibility barriers. Second is overcoming the seed dormancy and self-sterility of the seeds. Third, embryo rescue in distant hybridization where the endosperm development is poor. And fourth is it is used with the shortening the breeding cycle. Thank you.